Okay, so welcome to this next video in which we are discussing angiogenesis in chronic inflammation. Okay, so we've seen that macrophages and T lymphocytes start releasing angiogenic factors such as vascular endothelial growth factor A, VEGF-A, and fibroblast growth factor 2, FGF-2. These diffuse to the endothelial cells. Specifically, they're going to work on the endothelial cells of the post-capillary venules, okay? And there, on the post-capillary venule endothelial cells, you have receptors for these angiogenic factors. So vascular endothelial growth factor A has the vascular endothelial growth factor receptor 2, which it's going to bind to. And fibroblast growth factor 2 has the fibroblast growth factor receptor 1, the FGFR1. Okay, right. Now, both vascular endothelial growth factor A and FGF2, they are both these dimers, basically. They exist as dimers. So, when you release vascular endothelial growth factor, basically, if I get this picture, it actually consists of two vascular endothelial growth factor A proteins. Okay, now one of these proteins in the dimer will bind to one vascular endothelial growth factor receptor 2, and the other will bind to another, a second vascular endothelial growth factor receptor 2. Similarly, for fibroblast growth factor 2, again, it's a dimer, and one of the fibroblast growth factor 2s will bind to a fibroblast growth factor receptor 1, and the other will bind to a second fibroblast growth factor receptor 1. So both of these trigger the dimerization of their respective receptors. Then what happens is a process known as cross-phosphorylation. So the tyrosine kinases on the intracellular aspect of these receptors, so the VEGFR2 receptors, and also the fibroblast growth factor receptor 1, the tyrosine kinase domain will become active on the intracellular domains of both of these. And then what will happen is that this tyrosine kinase on one of these receptors that's in the dimer pair, so for instance, if we take the example of the vascular endothelial growth factor receptor 2, we have two vascular endothelial growth factor receptor 2s dimerized together now, which will label the first and second of these two receptors. And basically, the tyrosine kinase domain of receptor 1 will phosphorylate the tyrosine residues on the intracellular aspect of receptor 2, and the tyrosine kinase domain of receptor 2 will phosphorylate the tyrosine residues on the intracellular domain of receptor 1. Okay, so you'll then get these phosphotyrosine residues on the intracellular aspect of both of these receptors. Now, what the phosphotyrosine residues do are they recruit, many different effector proteins, and we're going to activate 3 different downstream pathways, 2 of which we've looked at now. So the first one that we looked at was this recruitment of a Ras-Gef complex, so the recruitment of a Ras guanine nucleotide exchange factor complex, which would then activate this Ras-G protein by exchanging the guanosine diphosphate that the off state of the Ras protein has bound to it for a GTP, turning this Ras protein on. That Ras protein then activates a series of serine threonine kinase enzymes, which leads to the overall activation of 2 transcription factors, MYC and CFOS-C-Jun. And we've said we're going to look at these transcription factors and what they do later. Okay. We then saw another pathway which involved the activation of phospholipase C-gamma. Okay, now phospholipase C-gamma is an enzyme that's usually within the cytoplasm of the cell. However, when you've got these phosphotyrosine residues on the intracellular aspect of these receptor tyrosine kinases, then phospholipase C-gamma will come and bind to those, and that will keep it nicely at the membrane, so you get migration of the phospholipase C-gamma from the cytoplasm to the membrane, basically. And now, phospholipase C-gamma will start breaking down phosphatidylinositol 4,5-bisphosphate molecules that are in the cell membrane into diacylglyceride molecules and also inositol 1,4,5 trisphosphate molecules, IP3 molecules. IP3 will then go and activate the IP3 receptors, and it will lead to the opening of the IP3 receptors, although IP3 binding doesn't in itself cause the opening, but it does lead to the opening of the IP3 receptors. Okay, so to keep it simple, we'll just say when the IP3 goes up, it leads to the opening of these IP3 receptors. This leads to an increase in the calcium level within the cytoplasm of the cell. So calcium level in the cytoplasm is usually kept ridiculously low at around 100 nanomolar. Now that is tiny, okay? If you compare that to, for instance, the intracellular potassium concentration, which is around 155 millimolar, then you can see how tiny that is. Okay, so this is around 100,000 times smaller, 150,000 times smaller than the potassium concentration. Okay, so in fact more than that, it's 1.5 million times smaller than the potassium concentration, so it's very, very small. Okay, right. So when you release calcium from the endoplasmic reticulum, then calcium will go up in the cytoplasm, and what this is going to lead to is it's going to lead to the conversion of apocalmodulin proteins into calcium calmodulin complexes, because the calcium will bind to the apocalmodulin, and that will convert the apocalmodulin into these calcium calmodulin complexes. Calcium calmodulin complexes will then bind to an enzyme in the cytoplasm of the cell known as calcineurin, or CAN for short, and calcineurin is then what's known as a serine-threonine phosphatase, and it removes phosphate groups from the serine-rich region of the NFAT transcription factor, which stands for the nuclear factor of activated T cells. Okay, now these phosphate groups on those serine residues within the serine-rich region, those were inhibiting the NFAT enzyme, the NFAT transcription factor. They were stopping it from being able to translocate into the nucleus. Okay, so once you remove those phosphate groups, the NFAT can move into the nucleus, and then it can activate, it can affect transcription of genes, basically. It can act as a transcription factor. And it's going to do the same thing, or at least a similar thing, to MYC and C-Fos-C-Gym. Okay, so we now want to see one final pathway which these receptor tyrosine kinases can activate, which will lead to the activation of another transcription factor. Okay, right. So let's start this off. So we'll draw our receptor tyrosine kinase dimer again. So here is the phospholipid bilayer of the cell. Okay, here is the membrane spanning regions. And extracellularly, you then have these immunoglobulin-like domains. And again, we'll draw the fibroblast growth factor receptor 1, simply because it has slightly fewer immunoglobulin-like domains extracellularly, so it takes slightly less effort to draw. Okay, so these are the immunoglobulin-like domains. We then have our fibroblast growth factor 2 dimer in the middle there. So let me colour in the fibroblast growth factor 2 dimer. So in purple here, these are the fibroblast growth factor 2 dimers. Okay, so this is fibroblast growth factor 2, FGF2. Okay, then we have the transmembrane region, and intracellularly we have this split tyrosine kinase domain with this kinase insert between the two, and then a tail after the second of the two domains of the tyrosine kinase. And then the same for this fibroblast growth factor receptor 1. Okay, split tyrosine kinase domain, and then a kinase insert there. Okay, so let's just label up the pieces here. So this is the kinase insert. These are the immunoglobulin-like domains, which often people write IG, and then they put like domains. So I, and then a little g like that, means immunoglobulin. Okay, right. So then what's going to happen is when the fibroblast growth factor 2 binds to this fibroblast growth factor receptor 1, then what it's going to do is it's going to activate the fibroblast growth factor 1. So this is the fibroblast growth factor receptor 1, okay, and it's going to lead to the activation of both of these tyrosine kinases. So this tyrosine kinase here is going to become active, and this tyrosine kinase here is going to become active, and of course they're going to cross-phosphorylate each other's tyrosine residues. And the tyrosine residues that they cross-phosphorylate are mainly in this kinase insert and also this tail after the second of the tyrosine kinase domains. Okay, so what are these phosphotyrosine residues now going to do? Well, basically they're going to recruit another enzyme known as PI3 kinase. Okay, so where should I draw this? This is kind of awkward, I've run out of space now. So I'll draw it attached to the phosphotyrosine residues again. Okay, so this enzyme is often abbreviated to PI3K. Okay, and this stands for phosphatidyl inositol, which we know is what PI stands for. Phosphatidyl inositol, and then free kinase. Okay, free kinase. So that's what PI3K stands for. And often people will just abbreviate this to PI3 kinase. So often you won't hear people read this as PI3K. Instead they'll read this as PI3 kinase, meaning PI standing for phosphoinositol and then free kinase. So just like with the MAP kinases, people don't read it as MAPK. They read it as MAP kinases. Again, PI3K we read as PI3 kinase. Okay, right, so we will continue this discussion in the next video, in which we'll see what this PI3 kinase is going to do.